The sliding filament model describes the process used by muscles to contract. It is a cycle of repetitive events that causes actin and myosin myofilaments to slide over each other, contracting the sarcomere and generating tension in the muscle. The contraction begins with myosin head binding to actin on the actin filament. The myosin head flexes, pulling the actin filament along with it. This causes the actin filament to slide by the myosin filament. Cocking occurs where myosin head is released from the actin with the help of ATP. ATP is turned into ADP in this process. This frees the myosin head to bind with a different actin molecule farther up the actin filament. The entire process may then be repeated many times. The structures involved in the sliding filament model are myofibril, a cylindrical organelle running the length of the muscle fiber containing actin and myosin filaments; sarcomere, the functional unit of the myofibril divided into I, A, and H bands; actin, a thin contractile protein filament containing active or binding sites; myosin, a thick contractile protein filament with protrusions known as myosin heads; tropomyosin, an actin-binding protein which regulates muscle contraction; and troponin, a complex of three proteins attached to tropomyosin.